and welcome to today's webinar. My name is Sherlyn Chen from Boehringer Ingelheim, and I will be your moderator for today's webinar, which is organized by the Gastroplus user group. Today's presentation is titled Development of a Unified Dissolution and Precipitation Model and its use for the prediction of oral drug absorption. This presentation will be given by Dr. Ruben Alvarez-Sanchez from Roche. Dr. Alvarez studied chemistry at the European Chemistry, Polymers, and Materials Institute in Strasbourg, France, and got his PhD in bioorganic chemistry at the University of Strasbourg. In 2003, he joined Hoffmann La Roche in Basel, Switzerland, as a postdoctoral fellow in the area of drug metabolism. From 2006 to 2012, he acted as DMPK expert in research project teams, being responsible for the coordination of ADME and PKPD activities from target assessment to clinical candidate selection. Since 2012, he leads the pharmaceutical profiling group responsible for the early characterization of compounds in the area of physical chemistry and in vitro safety pharmacology. Also participating as the panelist today is Dr. Vera Lukchova from Simulations Plus. Vera is the team leader for simulation technologies and also serves as the Gastroplus product manager. Vera will be available later on to answer questions related to the Gastroplus program. Following the presentation, there will be an opportunity to ask questions. You may either send your question using the question pane on your control panel, or you may ask your question directly using the hand-raising icon. If you are using the telephone to listen to the call, please be sure to enter the unique audio pin displayed when you join the call. This enables us to unmute your line so that we can hear you. This webinar will be recorded and it will be available to the user group members in the future. So without further ado, we now begin the webinar presentation. Ruben.
So thank you, Sherlyn, for the introduction. And also thank you to the organizers for letting me present this work here today. So the intention of this hour is to raise your awareness about what we believe is a quite interesting piece of work that can offer a great potential for the area of PBPK modeling, and in particular in oral drug absorption. So to start really at a higher level, the common reason we are all here today is because we are committed to find the path to safe and efficacious drugs. And to do that in a way or another, we get confronted to similar type of paradigms, and usually we get confronted, particularly for those working in early stages, to two key questions, which are basically how my in vitro data can predict the in vivo outcome, and also to what extent the animal data can be translated into a human profile. So probably I don't need to convince anyone in this audience that to do that, basically PBPK offers an ideal setting, because it helps us to integrate the drug-specific properties, so properties that we can predict in silico or we can determine in a bench, find this with information that we have in terms of animal or human physiology, and ultimately help in explaining or predicting the drug's PK/PD profile. So as part of this framework, one of the key processes we aim to understand is the oral drug absorption, which relates basically at how the compound properties can relate to systemic exposure. And we know that this is a very dynamic process. We also know that it's highly governed by compound physiochemical properties. And we also have a good understanding, I mean, the human and the animal physiology for the gastrointestinal tract is fairly well established or understood. And also importantly, it's relevant for a majority of drugs that are given via this route. And we also know that for a drug, in order to be absorbed, there are a certain number of key steps that the drug needs to overcome in order to get absorbed. And among them, dissolution and precipitation are really central. And those are the ones that we'll discuss about during this seminar. So when we look back at the dissolution theory, we see that its study started already something like 100 years back, and actually was not something intended for the pharmaceutical research, but it was more as a general physical phenomenon. It was actually many years later that this dissolution was actually related to oral bioavailability of drugs. But it was Noyes and Whitney back in the 19th century who empirically found that the rate at which a solid substance dissolves is proportional actually to the difference of the concentration in that solution and the concentration of the saturated solution, or the solubility, something that is somehow reflected on this empirical formula. So it was slightly later that Nernst and Brunner proposed a diffusion layer model that allowed for a more mechanistic understanding or explanation of the dissolution kinetics. And this was done on the basis of the solution of a solid particle with a certain surface area, the released molecules diffusion across a diffusion layer of a certain thickness, of a certain diffusion coefficient, and the molecules being then released into the bulk fluid where the concentration would increase up to a certain limit that was this solubility limit that is determined somehow empirically. So now, and in the course of the following years, besides some adjustments that are mainly mathematic and that are related to the way we can account for the surface area of a particle a long time, it's certainly fair to say that the Nernst and Brunner's equation continues to be the basis for drug dissolution modeling. And that's actually a quite interesting relationship in the sense that it's simple in its mathematical form, and it provides a mechanistic sense for the process. It's probably worthwhile mentioning that although precipitation and dissolution could be considered two sides of the same coin, dissolution is the only part that this equation actually reflects. And when we go into Gastroplus and we look how this process is captured, we see that there are a certain number of models that have been proposed and that are available and basically relate mathematically to this original equation. And as I said before, taking into account some modifications to consider the evolution of the particle surface during the time. So precipitation in its hand is a process that has been studied in pharmaceutical sciences to a much lesser extent, and it is known to be of particular importance, particularly in the case of weak bases. In fact, a weak base is expected to be highly dissolved in the gastric pH. At acidic pH, these compounds are fully protonated, and as the drug passes into the intestine, the pH becomes more alkaline, the compound becomes less protonated, and ultimately this relates to a loss of solubility that in some cases can lead to precipitation. So this phenomenon has been linked to exposure differences in populations with elevated gastric pH, and it is also known to be a potential source for drug-drug interactions for co-medications involving gastric pH modulating agents as proton pump inhibitors. So when we look at the available in vitro tools to study precipitation of drugs in biorelevant conditions, actually the most interesting literature that we find comes from the last 10 to 15 years. And one of the approaches that is followed has been to use some sort of static models where a drug can be dissolved in a gastric relevant medium, and after some time that can be representative of the gastric emptying rate, the resulting solution is then diluted or reconstituted with intestinal biorelevant medium such as FASIF. And here again, the concentration time profile can be followed over a certain period of time that could be reflective of the intestinal transit time. So in the example provided here, which comes from a publication from some BMS colleagues, they actually show that a weak base like gefitinib is able to dissolve very rapidly in this gastric acidic pH, and once the environment is switched to the intestinal more alkaline conditions, we can see that the compound precipitates very rapidly. So in a similar experiment where the pH in the gastric phase is increased, we can see that indeed this dissolution is slower and also is incomplete, and the dissolution continues also in the intestinal passage. So besides many things that were shown in this paper, the authors started or attempted some in vitro and vivo correlations using this data. And basically what they defined is the ratio of the AUCs of these different in vitro studies, one reflecting the normal physiology, the other one reflecting the elevated gastric pH physiology, and calling this the in vitro AUC ratio, and saw how this would correlate with the AUC ratio or Cmax ratio coming from clinical studies, drug-drug interaction studies in combination with pH modifying agents, and seeing that in some instances this could lead to interesting correlations. So another possibility is to use more dynamic systems, and in this regard, likely the most established model is the transfer model published by Kostevich et al. This was in 2004. And in this model, a gastric-like solution, here depicted in this donor phase, is pumped into an intestinal surrogate buffer, and this is done at different transfer rates defined within a pump. So what the data of this model shows is how the transfer of the compound in the intestinal vessel leads to a buildup of concentration, and in certain instances, particularly in the passive conditions, and depending also on the transfer rate, there's a point at which the system collapses and the concentration decreases down to the equilibrium solubility. So there are more complex models that have been reported aiming to recapitulate more subtle aspects of the gastrointestinal physiology. One of the examples is this mini gastrointestinal simulator depicted here, that, as we can see, is in a certain way a more sophisticated version of the transfer model. Or we have even more complicated models, such as the well-known TIM1. So these models have shown their usefulness in addressing specific absorption questions. However, given the high experimental complexity and API requirements, they are precluded in early phases discovery. On the other hand, the high degree of experimental deconvolution prevents a direct import of the experimental outcome in PBPK tools. Basically, we cannot use the outcome of such models directly as input parameters in our PBPK software. And maybe a bit provocatively, these models may raise the question of how complex a model needs to be in order to be predictive. And now if we look at the mathematical models available to deal with precipitation processes, in contrast to dissolution, this is clearly an area that it would be fair to say that continues to be work in progress. One of the assumptions that people have often been made is that the precipitation follows a first-order kinetics, which is directly proportional to the degree of supersaturation of the solution, or the difference between the concentration and the actual solubility. So when linking this type of model to in vitro data, it is actually seen that the rate constant that can be derived is actually not that constant as one would think, and it actually depends on the experimental conditions that are picked, mainly what is the starting conditions in terms of supersaturation, or what is the transfer rate used in the experiment if we are using the transfer model for studying this process. So a question that may rise is what is the actual precipitation rate that needs to be used out of these in vitro studies to predict the in vivo. So as you likely know, this first-order precipitation model is implemented in Gastroplus since long, and the way the default precipitation constant has been defined is by looking at the dypiridamole data produced in the Kostevich transfer model, and see how the precipitation rate constant, in this case expressed as precipitation time, varies at different transfer rates in the model, and then picking the transfer rate that would be in agreement with the flow of fluid that we find physiologically. So that's the way these 900 seconds that you may have seen in Gastroplus are actually derived and used in the modeling. So somewhat more developed precipitation model is the classical nucleation theory, and this has been recently added also to Gastroplus. So this model relies in thermodynamic considerations, basically about when a cluster of molecules starts to be thermodynamically more favored and can be formed. And being mechanistic in nature, this model shows, however, a high degree of mathematical complexity, and it requires in its formulation a certain number of parameters that many of them may be challenging to get out of a lab. So as a result of this complexity as well, the model is highly computationally demanding, and when it boils down to assess its relevance, there's little work done in terms of validation. Actually, most of the work relates to the work done by Sugano to model the very same transfer data that I showed before about dypiridamole, which, as you can see, it can be nicely achieved when fitting a couple of parameters to the model. So this slide shows, in a nutshell, what are the equations involved in the model and the different parameters or inputs that are required. I mean, my intention here is not to explain it, something that probably would be a little bit far away from my own ability, but just to stress the points that I made before about complexity and disconnection from a lab bench. I also leave here a couple of publications in case of anyone interested. So a bit as a summary, what is the model toolbox that Gastroplus offers to cover these dissolution precipitation processes? So actually, Gastroplus handles solubility using different models, and it in principle extrapolates solubility across pHs according to the compound's degree of ionization using a modified Henderson-Hasselbalch model. And to do that, Gastroplus can rely on solubility data given at a single pH, and this can combine with the so-called solubility factor, which is basically the ratio of the ionized and no ionized forms of the drug. And together with this, it's also possible to correct the solubility at different bile acid concentrations using the Michanis equation, and then link this solubilizing effect into the model by taking into account what are the available physiological levels of bile acids across the different GI tract segments. So as far as dissolution is concerned, Gastroplus offers, as mentioned before, a set of dissolution models, as well as the ability to deal with more complex polydispersed systems. So regarding the precipitation part, as just mentioned, it offers a couple of models, the first order precipitation model, as well as the more complex classical nucleation theory. So as we can see, G+ already offers a quite comprehensive set of models to deal with dissolution and precipitation. The question that we may have, and actually we had, is there a need for anything else? So we started out our work from the realization that in early stages of drug research, the first human dose predictions typically rely on very thin biopharmaceutical data packages, which may differ from company to company, but often at Roche, they may rely in equilibrium solubility data in biorelevant media. The reasons for this are mainly high compound demands and large experimental requirements needed to generate dissolution or precipitation data from experiments, which is often not compatible with a large amount of compounds that we study in early stages. So on the other hand, we also thought that the current models for intestinal drug precipitation are actually too simple or even simplistic or fairly complex or over-complex. And for the latter also, they may not be directly connected to the experimental methodology that one could expect to have in a regular physics lab. And this is how we identified the interest of providing some earlier access to more comprehensive dissolution and precipitation profiling by means of small-scale methodologies, and ideally bringing this experimental output, bridging it to PBPK modeling. So the way we thought we could go about it is first to establish an in vitro methodology to study the dissolution and precipitation processes. This means basically for early stages experimental tools with appropriate throughput and low compound needs. Also then to develop a mathematical framework that could allow for a mechanistic explanation of the in vitro data that we generate, and ultimately bridge this experimental output to in silico approaches, and ideally to approaches such as PBPK. So for the part that relates to the in vitro methodology, we were interested in exploring the possibilities offered by the Sirius T3 platform. Actually, we had some extensive and positive experiences using this machine in our labs, mainly for the determination of high throughput pKa's. And we wanted to explore its potential for studying dissolution and precipitation. So what makes this instrumentation interesting for us is that it is highly automated, it requires very limited amount of API, and importantly, it allows for real-time monitoring of the concentration time profile for the experiment, with no need of sample workup procedures or any type of offline analytics. So the experiments are run in a very small vial, actually 2 to 3 milliliter vial, in which it's possible to fit a bunch of different devices and sensors. So we can fit a stirrer, we can also fit a UV probe, we have a temperature sensor that helps us to control temperature, and we have also a pH electrode that helps us to control the pH. And on top of this, we can also flexibly add different type of buffers, acids or base that can help us to dynamically change the buffer composition and pH. And the type of experiments we run were actually classical pH shift experiments, similar to those that I presented before. So in essence, what we started, we started from a fully dissolved weak base that is dissolved in gastric pH conditions, and eventually we induced precipitation by changing from the gastric to intestinal biorelevant conditions by adjusting the pH and adding the necessary ingredients for that in order to get some sort of passive surrogate. So under these conditions, it was possible to see how the compound was fully dissolved in this gastric phase, and once the buffer is changed to intestinal conditions, we can see then how the compound precipitates. So we tried this experimental setting for a large set of weak bases and actually found out in the process that for the big majority of them, the light scattering induced by the precipitate prevented the acquisition of useful profiles. Nevertheless, we were lucky enough to come across some few compounds where we could obtain neat data and really have sufficient data to continue the work. So we ran such precipitation studies for those compounds starting at different degrees of supersaturation. And we're also interested in looking at the concentration time profiles produced in passive and also in blank buffer. So this means same pH but in the absence of bile salts and phospholipids. Here, in order to see whether there could be potential differences due to the formation of micelles. So the data that we obtained that is shown here shows that in principle the compounds, irrespective of the buffer, followed a very similar type of behavior and very nicely recognizable patterns. So we could distinguish what we call a latency time, during which the compound can stay in supersaturation state in solution, and this is actually followed by the so-called precipitation phase that happens more or less quickly. Actually, what we observed is that the higher the supersaturation in the experiment, the shorter the latency time and the quicker this precipitation phase. And after precipitation, the compound reaches an equilibrium concentration, which is actually the equilibrium solubility. We also found cases where the concentration would stay above this equilibrium solubility during the full experiment. And we're interested in following these things up and found out that the equilibrium solubility can also be reached to those experiments provided that we wait long enough, meaning waiting long enough could be as long as 1 or 2 weeks for some cases. We also found that it was possible to induce this equilibrium in a faster way by adding some silica gel particles, for example. So if we go back here, we see that this is a pretty much a concordant pattern and behavior that we found. And this was in a way something that made us believe that there can be the chance to do something about it in terms of mathematical modeling. And this could help us also in understanding better what's going on and importantly leveraging the usefulness of this data for predictive purposes. So that's why we decided to do the maths, and to do that, we decided to take some sort of reaction kinetics type of approach. So what we actually proposed is that the dissolution precipitation process for a compound can be decomposed on the combination of a certain number of elementary reactions or reaction steps, if you wish. So these steps could actually be the dissolution, which is basically the release of a dissolved molecule out of a solid aggregate. This would be also particle growth, which would be the addition of a dissolved particle onto an existing aggregate. And nucleation, which in this case would be the association of a certain number of dissolved molecules to form an embryonic solid particle. So to make things simple, we assume that the compound can be either dissolved or undissolved. And the interconversion among them could be expressed on the basis of these three elementary steps. So without going too deep in the maths, what we see is that we have a dissolution term that's basically proportional to the rate constant of this step and the amount of solid, which is actually the surrogate for the effective surface. And we have also a growth term that is basically the result of the product of the rate constant and the reactants. So this is solid and dissolved material. And we have a third term nucleation, that in this case, again, is the result of the product of the rate constant, the reactants exponent, the molecularity index, and this in this case is scaled up to the total volume of the vial. So at the end, what we have is a system of two differential equations, which are basically the same but of different sign, and that account for the variation of the dissolved amount and the solid amount. And these two differential equations are parameterized with three different rate constants and this molecularity index for the nucleation step. So with this model in hand, we went through the fitting exercise, and here we found that the entire data set for a single compound in a same buffer could be nicely described on the basis of such model, and in this case using the exact same parameter set. So in other words, we perform a simultaneous fitting of the different precipitation profiles, and we managed to describe all these different profiles on the basis of a single set of 4 parameters for a same buffer, same compound. So here in the slide, you can see a couple of examples of the goodness of this fitting. And as we can see, we can indeed recapitulate the different curve patterns that we mentioned before. So we also analyzed the model a little bit further and found that the nucleation rate is a very slow process when we compare it to the dissolution and growth rates. And actually this term is important when we need to describe the initial part of the precipitation, that is when there is no solid available, but once a limited amount of solid becomes available, the term can actually be neglected. So if we neglect this term, and we also postulate steady state conditions, we obtain the following relationship. So this means no change of concentration over time, and this means that the concentration in the solution corresponds to the equilibrium solubility. What we found is this pretty much interesting relationship, which basically tells us that the equilibrium solubility is the result of two counteracting kinetic processes, which are particle dissolution and particle growth. Also, if we take this relationship and plug it into this initial equation, taking out already the nucleation term, we can rearrange the equation and make it look like this, which, as you may have noticed, it looks pretty much comparable to the Nernst and Brunner's equation, which basically shows how this dissolution theory, postulated longer than a century ago based on Fick's law, could in a way be reconciled with this dissolution precipitation kinetic theory, and also importantly, that this model can be simplified to an extent where instead of having 4 parameters, we could have 2 parameters, and we could basically identify these parameters using the classical dissolution experiments. So as mentioned earlier, the model proposed here relies on the assumption that the system is monodisperse and the surface to mass ratio is constant along the experiment. Of course, in an experiment involving particles ranging from embryonic nuclei to macroscopic aggregates, this may sound somewhat unrealistic, and is certainly an aspect where additional complexity could be added to the model. So, however, this assumption offers the advantage of reducing the mathematical complexity and the computational demands. Actually, a model like this could be very easily run in a simple spreadsheet, and it could be thought that such a model could also be linked to polydispersion models already available within Gastroplus. So alternatively as well, it is possible to somehow experimentally bias the model towards conditions that are more relevant for the study of the drug. In other words, if the in vivo conditions that we aim to model or we anticipate involve mainly drug dissolution, it might be good to enrich the dataset with dissolution studies. In contrast, if we anticipate intestinal supersaturation conditions, it may be better to design experiments that cover more the precipitation part, so that at the end of the day, when we do the fitting to get the 4 parameters that characterize the system, those parameters are to a certain extent biased towards the type of data we aim to model. So at the end of the day, what is important is that we try to keep the model simple, but ideally not simplistic. So one of the aspects we got interested in is on the influence of the undissolved compound on the precipitation kinetics. Actually, when we look at some published precipitation models, we often see that the precipitation rate is linked only to the concentration in the solution and not to the amount of solid that exists. However, when we look at the formulation of our kinetic model, actually the solid amount is one of the terms that is taken into account and can be seen that it could have an influence in the precipitation kinetics. So in order to address this question, we thought about an experiment where we could basically induce the supersaturation and look at the precipitation kinetics after addition of a given amount of solid material. So in this slide, you can see the influence of the addition of undissolved dypiridamole on its precipitation kinetics. So the experiments start at a certain degree of supersaturation concentration for dypiridamole. Actually, in this case, we designed 2 different experiments at 2 different initial concentrations. And what we can see is that the rate of precipitation or the precipitation kinetics in general is significantly accelerated as we add increasing amounts of solid into the system. So we also wanted to see how those results could be explained using the model parameters that we obtained in our pH shift experiments that I showed before. And actually, we were very pleased to see that the parameters that we defined before in our former experiments were able also to explain this type of data. So this phenomenon could be of particular relevance in vivo in cases where we have a weak base that is not fully dissolved in the stomach and that is partially transferred into the intestine as a solid. In such cases, it may be possible that the undissolved material can act as some sort of catalyst, and this can induce or accelerate a faster precipitation process. So important just to say is that indeed the amount of solid material can influence this precipitation kinetics, in contrast what is postulated, for by simpler models like the first order precipitation model. So we decided to go to step further and check to what extent the model could also explain other existing data sets. And actually we were particularly interested in the transfer model, since this data published by Kostevich were also generated using dypiridamole, one of the compounds that we run in our experimental set. And this is a well-recognized model that has also been attempted to be modeled before by Sugano using the classical nucleation theory. So what we wanted to check is how does the model describe this dypiridamole data. So we extracted those data, particularly the data in FASIF that were published, and we slightly modified our own dissolution precipitation model to take into account the experimental conditions of the transfer model, namely the supply of the compound at a given transfer rate. And for this exercise, again, we used the parameter set that we obtained from our pH shift experiments. In other words, we did not try to fit the data as done in Sugano's work, but we actually tried to explain or predict the data of this experiment based on our own data and model. And in this graph, you can see how the experimental data and the model overlay and how the simple pH shift studies and the model can explain more complex models such as this one. So this brings me back to the question I dropped before about how complex a model needs to be in order to be predictive. So these results encourage us very much in getting to the next level and try to predict oral absorption. And in order to do that, we designed a very simplified physiological absorption model involving, in principle, a stomach compartment characterized by the first-order emptying rate, as well as an upper intestinal compartment characterized in this case by an emptying rate, a surface derived from its average radius, and a volume of fluid. So within this upper intestinal model, we incorporated our dissolution precipitation model that would account for the interconversion of solid and dissolved material, assuming in this case that the compound that is transferred into the intestine comes all as dissolved. So the parameters used in this model were directly extracted from the Gastroplus ACAT model for human fasted state physiology, and as for the permeability that is also needed, we obtained it from the ADMET predictor software. So from the modeling done, it's already possible to get insights into the coexistence of the different pools of compound, dissolved compound, undissolved, and absorbed, similar as we do for Gastroplus. And from such profiles, we can see here, for example, in the example of erlotinib, that the model suggests a very quickly increase of concentration dissolved as the compound enters the intestine, and at some point the system collapses and the concentration drops to the equilibrium solubility concentration, and this further goes down as the compound leaves the intestine. So we also wanted to see to what extent the model would be able to explain the fraction absorbed for those compounds where for the cases where we had compounds where the fraction absorbed could be calculated based on clinical data. And we could see that we also find a relatively good prediction when we compare to the clinical data. I must say, and this is probably a bit apologetic, that every time that I show this graph, I'll feel a little bit embarrassed, as this graph appears to be suspiciously too good. It may be worth saying that even the numbers being close, the uncertainty that can be associated either to the estimation of this fraction absorbed or to the prediction made with this simplified model are big enough to probably put this exercise rather as a qualitative than a quantitative exercise. Nevertheless, it's great to see that this type of approach helps to rank order compounds in the right way. So we're also interested in addressing those proportionality questions on the basis of this model. And to do that, we performed also sensitivity analysis and see how increasing the dose will actually impact the fraction absorbed or also the amount absorbed, and how this would be translated also into a certain increase of the fraction precipitated. And we were also interested in what extent the expected clinical dose would fall into this sensitivity analysis. Of course, thinking that an ideal drug, we should have a relatively unsensitive behavior for the amount that is absorbed over the dose range that is intended for the clinical trials. So we also ask ourselves whether, besides being able to predict fraction absorbed, the model could also be able to explain the actual PK profile, provided, of course, that we have the knowledge on the disposition parameters. And we're also interested in knowing to what extent the model could extrapolate to other physiological conditions. And to address these points, we performed a compartmental analysis, PK analysis, and replaced the absorption rate constants determined in this analysis by the simplified absorption model that I showed before. So afterwards, we compared the experimental data and the predicted PK profiles. So to put some images into these thoughts, what we basically did is to perform a compartmental PK analysis and determine the different disposition parameters. And once this done, we basically took out the part that relates to the absorption and plugged in this simplified absorption model, keeping constant the disposition parameters that were previously fitted. And we actually applied this model and compared with the available clinical data. And we were particularly interested in model a complex case, the case of erlotinib, where a recent drug-drug interaction study in co-administration with a gastric pH modulating agent, omeprazole, had been recently published. So to model this study, we used our pH shift data to model the arm receiving only erlotinib. And for the cohort that was treated in combination with omeprazole, it is expected that the compound dissolves slowly and incompletely due to the elevated gastric pH. And thus we parameterized the model with dissolution data, dissolution particle growth rate constant that were obtained actually from dissolution data at the relevant pH. So as you can see, the model was able to capture quite nicely the absorption profile, matching Cmax, Tmax, and AUC. And actually, all in all, this work provided enough evidence to us that this framework can be a useful framework to model oral drug absorption. So now changing gears a little bit, we thought that the model could also be used to explain solvent-mediated polymorphic transformations. So to our knowledge, so far, hypothetical polymorphic transitions occurring in vivo have not been addressed and certainly not captured within PBPK. So at the moment, the model that we have proposed actually describes the interconvention between dissolved and undissolved compound, assuming that the undissolved compound is a single solid that is characterized by 4 parameters. So one could think about an extension of the model. And in this case, instead of describing only one solid form, we would have actually a set of solids, each of them characterized by a different set of 4 parameters. And at the end, these different solids could influence the amount that is in solution. So if we look at it in graphical terms, this would mean that the compound that is available in dissolved form could nucleate and could aggregate. And this could happen giving rise to 2 different solid forms. And actually, these 2 solid forms could actually also interconvert to from one to another, this doing it via the pathway of dissolution and reprecipitation. So this sounds a priori a quite reasonable mechanism. Of course, the first question that we had is how would theoretically this model behave? And here in this, you can see a time profile for a situation where a compound could be found in supersaturation conditions and could lead to formation of 2 different solids. So what we see in the profile is that the dissolved compound first precipitates, and one of the solids, the one that is kinetically favored, is formed predominantly. So this leads to what we call a pseudo-equilibrium or pseudo-steady state, where the solute concentration is determined by the solubility of this solid. So that's this level here. And the relative amount of the different species remains apparently constant. But during this time, the other solid form starts to build up progressively, and after some time, its formation rate starts to accelerate. So this leads to a neat interconversion of the 2 solid forms, and as this happens, also there's a decrease in the concentration of the solute, and this gives at the end rise to the final steady state, where the more stable form is quantitatively formed and the kinetic form disappears, and we have a shift in solubility from one to another. So this is theoretically what the model would postulate to happen. We're of course deeply interested in finding out what is the real physical behavior for those systems. And we couldn't find any example in the literature for in vivo polymorphic transformations reported. And actually, I would be curious to know if any of you may have come across such data set, because I think that this is really very nice data to model. We nevertheless found some few examples of polymorphic transitions monitored kinetically in different experimental settings. And in this case, the data showed here corresponds to the transformation of L-glutamic acid in water at 45 degrees that was reported in a quite interesting article. So the experiments, the dots correspond to the experiments, and actually the lines correspond to the fitting done with the model that we have proposed. And as you can see, actually the data is very nicely behaving the way that was anticipated, and the model very nicely captures this transformation kinetics. So just to wrap up, we have had an overview on the main dissolution and precipitation models that are available in Gastroplus, and we have seen a new combined dissolution precipitation kinetic model. So this model shows a number of interesting points compared to other models available. It's mechanistic and simple. It's actually linked to accessible in vitro outputs that we can generate out of the bench. Computationally manageable and implementable within PBPK softwares. It's been validated against more complex in vitro systems like the transfer model and also some clinical data. And it's also able to describe situations like solvent-mediated polymorphic transitions. Now, if I come back to the question I dropped before about Gastroplus toolbox, is there the need for anything else? One could think that a model like this could offer an interesting alternative and position itself as a unified solution to address dissolution and precipitation altogether. Probably as well, it could be linked to already existing polydispersity features that could also help in accounting for the variations in particle size. So where to find more? I can imagine that this has been a little bit of a tough dumping of a lot of concepts and ideas and data, and there is a quite large amount of questions that may come when it boils down to the details. So for those who are interested, I would encourage to refer to our recently published article, where you can find the vast majority of what has been shown here today and a little bit more. So for those interested and going through, you will find also data that has been shown here, and importantly also the model codes that you need to reproduce those data, and also for you to play around with your own data sets. And for that matter, I would certainly be glad also to know about your own experiences when dealing with it. So let me finish thanking all the people who have contributed in a way or another to make this work possible. People across the organizations and disciplines, mainly physiochemistry, formulation, modeling and simulation, pharmacokinetics. So I would like to thank also particularly our student Paulina, who did the hard work and probably is now asleep with her headphones on the head. So with this, I would also like to thank you all for following me till the end and willing to take any questions. Thank you.
All right, thank you very much, Ruben, for the great presentation. At this point, we will open up for questions. So I'll read the list that list of questions that came along while you were giving the presentation. The first question we have here is, can you define C1, C2, etc.? This is from Steve Sutton.
C1,
the
C2.
definition of C1, C2.
In what slide, may I ask?
The question did not specify which slide. See if I can.
I think it appeared before that slide, somewhere in the early slides before you were. It was before you started presenting your own model.
Mm-hmm. So is maybe that one?
I think it was later.
I see a 1, but not a 2.
Okay, so Steve just clarified it was on the slide titled Precipitation of Growth in Vitro.
Okay. It's in the slides. In the slides titled "The Precipitation of wet granulation in vitro."
I think it was before this slide that you were showing, the last one.
Which one do you mean? Sorry, Vera.
The one that you were at before. Yeah, maybe the one that you are pointing out right now. There is a little delay.
Excuse me. So maybe best is that we go back to the person asking the question, and he may give a little more of details.
Actually, Paulina is watching, and she said that it's slide 20.
20. Oh, thank you. This is slide 20. Oh, yeah. Thank you, Paulina. So yes, this refers actually to the initial concentration. I mean, to the initial concentrations on the experiments. So we call them by Cs, but in principle, they would refer to the absolute numbers that are reflected in the slide. So I hope that this answers the question.
Actually, Steve's comments, your slides are not numbered. He could not figure out what's the number, I guess.
Yeah.
Okay.
So basically, C1 would be this value here, and C2 would be this value here. And what this means is that each curve corresponds to different concentrations in the in vitro profiles, different starting concentration.
Different initial concentration.
Yeah.
Okay. The next question, how can you explain the sustained supersaturation?
Sorry, Shirley, could you repeat, please?
How can you explain the sustained supersaturation?
So how can I explain the sustained supersaturation? The latency time, I guess that this refers to, right?
How you define the sustained supersaturation?
Yeah, mechanistically. Actually, we looked at it in very much detail, in the sense that it's somehow surprising that a system like this can be sustained at a supersaturation level in a so stable degree, and then all of a sudden crash out. And for some instances, as I mentioned before, even cases where these supersaturation levels can be sustained for over weeks. And when you look at the model, what it tells you is that for the growth of the particles to take up, there needs to be a significant number of nuclei that is available to do this. So once you have enough nuclei, the rate becomes really exponential. But before that, you really need to rely on a very slow molecular process, or a process that happens with a low probability. And this means that for as long as this process is not enabled to generate enough nuclei, this means that you can get to a certain extent to a high supersaturation.
Okay, thank you. The next question. Precipitation of a drug due to crystal growth can be inhibited by encapsulation in suitable polymers. Can such a system also be modeled suitably?
So that's indeed, I guess, one of the next steps. What we did here refers basically to work done with either solid, or in case of precipitation, starting from fully dissolved compounds. For sure, the systems can become more complex as we start to think about formulations. And of course, the models become also more complex, and this needs to be taken into account. So I guess that this could be the basis for describing a particle at the end of the day. If you want to incorporate any type of technology on top of it, you will also need to get additional understanding on how this technology behaves kinetically, and importantly, how it behaves in physiological conditions.
Okay, thank you. The next question actually is for Simulations Plus. Does Gastroplus does Gastroplus take into consideration the presence or absence of the same condition, especially for BCS2 drugs?
Yes, so Gastroplus does take that into account, and as Ruben was showing some of the early slides on the dissolution models within Gastroplus, you would see that the dissolution is driven by the sink between, or by the concentration gradient between the solubility of the compound and the actual concentration of the compound in the intestinal lumen. So as the drug is being absorbed, the absorption and the removal of the drug from the small intestine or from the colon, wherever the absorption is happening, is going to be affecting that dissolved concentration and is going to be affecting that concentration gradient for the dissolution or for the precipitation process. So Gastroplus certainly takes that into account, and I believe in the PK simulation that Ruben was showing towards the end with his model, it was also taken into account because he had the permeability term accounting for, again, removal of the drug from the simplified gut lumen. Is that correct, Ruben?
Yeah.
Yeah, so both models, the one that's implemented in Gastroplus as well as the model that Ruben was using here to test this new precipitation model, would be accounting for the presence or absence of the sink conditions.
Thank you, Vera, for clarifying that. The next question, a comment regarding modeling of in vitro data, a second compartment mimicking tissue, such as an octanol, seems not to be relevant based on the data. It's a comment.
So can you repeat it, Shirley?
The comments about the modeling of in vitro data, a second compartment mimicking tissue such as an octanol, seems not to be relevant based on the data.
So I don't know if I got the question correctly and whether it refers to the two compartments that were postulated in the PK model.
Yeah, I'm not really clear about this question though. So it's just the second compartment mimicking tissue such as an octanol seems not to be relevant based on the data.
So if it is about the relevance of P2,
you do not? Yeah. Okay. That
so
might be the question.
If that's the question, sorry if it's not. Otherwise, you can also reformulate it again. Basically, we tried also different PK models with 3 compartments or single compartment, and based on parsimony, actually the 2-compartmental model was the one that was fitting best the disposition part of the curve.
Okay, thank you. You mentioned that T3, the TNO, T3 did not work for some of the compounds that you tested. Can you describe in detail what the issue was? Also, what was different with compound that did work on T3?
Okay, so I mean, I must say first that for us it was already great to have this platform available and generate some data with it. Of course, technical challenges arise, and when it's about it, the one that we were most confronted with is that the compounds, when they precipitate, they scatter light, and then the UV absorbance, which is our readout to determine the concentration over time, gets affected. So this usually translates into UV absorption profiles that are shifted up compared to the baseline, and that this is not a result of a true absorbance of the compound, but it's actually, the scattering effect of the aggregate of the solids that are formed. So in many cases, when this happens, we tried to do some subtraction or baseline adjustments, etc., but often you end up having data that is not usable. So that's for the part that relates to the technique. Of course, there were other challenges that were more related to, for example, what's the concentration range that we can study if we are using a UV signal as surrogate. We ideally need to have concentrations that are compatible with the linearity of the UV lamp, And yes, in that matter, we also had some cases where you start to test concentrations that are actually far away from this linearity range.
Okay, thank you. A question from Philippos regarding the measurements. You mentioned on slide number 19 that you only had luck with a certain set of the compounds to get these measurements, depending on precipitation. If the compound was to form small particles during precipitation, any suggestions on doing these measurements?
So, I mean, we did this on the Sirius because that's a very streamlined platform that is optimum for early work. I guess that if one wanted other compounds that do not work in this platform to work, one could either go to classical precipitation experiments with all the technical inconveniences that this means. Sirius Analyticals has also another platform, Infor platform, that has improved features, as I've been told, in the area of baseline correction algorithms, and maybe also for the part that relates to the technical acquisition. Something that may be of thought, and this is probably for the future, is having this same type of analytical techniques, not necessarily based on UV, but based on Raman, where in principle these things could be minimized. But yes, for practical purposes, if a compound didn't work in a platform like this, at present moment, if we had to work on a project and provide some data and generate some parameters, we would probably need to go into classical precipitation experiments.
Filippo also asked the second question. Do you have any experience with using this model for amorphous/crystalline modeling?
No, in the sense that we haven't come across with any data set where there's conversion of amorphous to crystalline. But let's say that these polymorphic, solvent-mediated polymorphic transitions that I alluded to before could also be thought to be modeled using the same mathematical framework for transformations between amorphous and crystalline, for example. So the same concept could still be valid. So this would mean in that case that the amorphous would be considered as an independent solid pool, and this solid pool would be characterized by another set of 4 parameters. But if you have this data, Filippo, yeah, maybe that's good. We can give it a try.
Okay, the next question is asking you, do you also have examples with weak acids?
No, we haven't done anything with weak acids. The reason for that being that we were interested in complex cases. And for us, the complex cases involved actually weak bases, which are the ones where we can see or we can anticipate precipitation in intestine. So we started with the difficult cases. I guess that these models could work with weak acids, could work also with other type of compounds. Of course, the type of data that would be needed would be different in the sense that we don't need to induce any precipitation, because that's something that we know is not going to happen. But we would probably need to supplement the model with data coming from dissolution experiments. And in that case, we could also work with a simplified version of the model that does not involve nucleation, but only involves dissolution and growth, which means that we could get basically to a very simplified model that resembles very much the dissolution equations that I showed at the beginning.
Okay, thank you. The next one is the in vitro model, which is the Sirius apparatus, capable to test the drug product, parentheses, capsules, tablets.
So in the setting that we tried, that is the one that we would consider ideal for our purposes in the lab, which are more focused to early stages. This wouldn't be possible because we would be dealing with very low amount of compounds that are usually below 1 milligram. So, but in this same platform, there's the possibility to work in a less automated fashion with bigger vessels, and it's possible to generate tablets with a press, and I don't know, probably you can also use capsules and see the release. So there could be possibilities of dealing with this type of, let's say, starting material. Also to consider as well, if that's the direction one wants to go, there are other platforms, as I mentioned before, like the Inform, that could be used for that purposes that maybe are more suited for this type of work.
Okay, thank you. Next question. Is it possible to expand supersaturation due to formation of relatively stable, more soluble polymorphic form? Normally, the more stable polymorphic form is not is less soluble, right? But anyway, this is the question. Is it possible to expand supersaturation due to formation of relatively stable, more soluble polymorphic forms?
So I don't know if I understand the question. So in principle, if we start from a, you say more stable polymorphic form, is this what I understood?
Relatively stable, more soluble.
More soluble, okay, so less stable. So if we start from a polymorphic form that is more soluble, it might be possible to reach an extra degree of, let's say, concentration in the lumen, and thereby increase the absorption rate. And then, provided that this form doesn't change very rapidly into the more stable form, it might be possible, not necessarily to reach supersaturation, but reach concentrations in the lumen that are higher than the ones that one would achieve with a more stable polymorphic form. Theoretically, yes. The question is, and that's more for the formulation colleagues, I mean, usually what is desired is to work with a polymorphic form that is stable enough, that is actually the most stable, so that we can avoid any type of hassles related to the production of the drug and the shelf life, which may be already a important enough reason not to think about these approaches, especially knowing that for many of these polymorphic transitions, the delta in solubility of the two forms is not that high that one would consider that this is so much interesting. I hope I have answered this question.
Thank you, Ruben. The next question from Neil. Possibility to model different polymorphism is very exciting. Could this also be applied for a dosed salt form, which precipitates as a free base with lower solubility than the salt form?
So can you repeat the question, Shirley?
Could this model also be applied for a salt form that's dosed, which precipitates as a free base, and the free base has lower solubility than the salt form?
Yeah. So, I mean, that's certainly something that one can think about doing. The question is, in those cases where you have a salt of a base which is most likely already fully soluble in the stomach, you would then basically deal with a single solid form in intestine, which would be the one corresponding to the precipitated free base. So in principle, the answer would be yes, you could model such system. And if the question is how, I would address this from the standpoint where, I assume that my compound is fully dissolved in the stomach, so the salt becomes really a dissolved compound, and then in the transition to the intestine, it precipitates to the corresponding amorphous free base. And then this would end up not being a polymorphic transition or anything like this. This would be more like the simple model where you would have a solid form and the dissolved compound.
Thank you. The next question is from Chen Ming, actually my colleague from BI. What is the physical meaning of alpha term in the nucleation equation? Will it be constant for different supersaturated concentrations? And I think the next question is, can Gastroplus simply add this option into current precipitation option so it can be accessible using the current Gastroplus model frame? There's a couple of questions here.
So the first one relates
physical meaning of the alpha in the.
So maybe I can answer to that one, and then I can hand over the second one to Vera. The alpha actually relates to the hypothetical number of molecules that would collide together to form a solid aggregate. So speaking, this really sounds like a little bit of a very unlikely event, but one could think that this alpha contains also, would incorporate in its meaning also particles that collide that are too small to be considered as incipient nuclei. But if we take the classical reaction kinetics approach, and that's the one that we took to make the mathematical formulation, this term is called molecularity index and basically accounts for the number of different molecules that come together to create the product.
Thank you. The next question, what is the time frame or how long does it take for the drug dissolution precipitation reaches steady state in your model?
Sorry, Shirin, there was a second question there about...
I'm sorry. Okay, yeah, I apologize for that. The second question is, were this parameter constant for different supersaturated concentrations?
I think the second question that Ruben meant was the addition to Gastroplus, which actually several of you have asked whether we would consider adding it.
That's the third one, but it can be considered second. Yeah. So the last question is from Chen Ming is, can this be simply added into the existing model already in the Gastroplus program?
Yeah. So I've seen that several of you actually asked that same question. We are certainly considering it. It's just a matter of timing. When we've seen this work, it does seem like a very intriguing work, showing a lot of potential. So we are certainly considering adding it into the program. It's just a matter of I just cannot give right now a timing when it will be available. We are in the middle of a bunch of different additions and modifications to the program right now, so we need to finish those up first since they are already in the works. But I think I can say with confidence that it will be added. It's just a matter of how quickly we can get to it and incorporate it into the program.
Thank you, Vera. Move on to the next question. What is the time frame? How long does it take for the drug dissolution precipitation reaches steady state in your model?
So, I mean, steady state can be reached at different times, really depending on what are the conditions for testing and what is and the compound itself. I mean, when we look at the data, basically these data are collected in intervals of 1 hour. And we can see that really, depending on the starting conditions, you can reach a steady state at different time points. And in some cases, as for example, this black line here, steady state is not reached until, I don't know, 1 or 2 weeks. So really, the time frame can be very different depending on the conditions. And that's for the part that relates to the in vitro. If now we go to the in vivo, of course, we have here the limitation that the compound is present in the intestine for a limited amount of time that is defined by the transfer rate of the compound out of the intestine. And in that regard, of course, a steady state would be found once the whole amount of compound is out and the concentration is 0 in that compartment. And then, of course, the question that it comes is how the time frame that the compound seems to have in the conditions that seem to be physiological is compatible with the time frame of the intestine to be emptied. So it could be that in certain cases, certain compounds, certain doses, the compound can live in a supersaturation state and be absorbed. Maybe in some others, as soon as it reaches the intestine, it precipitates quite quickly. I mean, just maybe a way to go about it is to show this slide. What I showed before, where you can see that, for example, in this case, the compound enters the intestine, it goes up very rapidly. At certain moment, this supersaturation is such that the compound starts to precipitate, and it does this very quickly. And the steady state is reached once the compound leaves the intestine. If we look, for example, at the case of dypiridamole, here there was no precipitation at all. And what we can see is that the compound can simply come and go from the intestine without having really a major impact in the precipitation, even though maybe the concentrations would be supersaturating concentrations.
Yeah, actually, Ruben, the person who asked this question further clarify, and this is in comparison with the GI transit time. Compared with the GI transit time. I don't know if that helps you to.
Mm-hmm. Yeah, so maybe that's what I was saying, that indeed in vitro can be one thing, and then in vivo can be another thing. And really depends how fast this happens in vitro and how, for certain conditions, how these conditions are actually reflective of what is happening in vivo. In any case, you know that you have a very well-defined time window where something can happen, and then it really depends on the, how high you dose and what are the conditions of the patterns or the behavior of this compound in such conditions.
There are several questions related to whether these slides or the presentation will be available later. I guess the answer is that it will be posted on the website of Simulation Plus. So it will be available later on. Is that correct?
Yeah, the webinar is being recorded, so the recording will be available just with any other webinar.
Okay. The next question, what are the analytical techniques that can be best used to determine the precipitation time in vitro, and what media is best suitable for use in modeling?
So for the analytical techniques, I mean, what we have done in this work mainly relates to UV absorption in this platform that I mentioned before, the Sirius T3. Whenever this doesn't fly because of the issues that we also mentioned a moment ago, then I guess that probably the way to go, and really for discussion, would be to go into classical precipitation experiments and then rely most likely on HPLC UV or HPLC MSMS. And there was a second part of the question, right?
No, this is just one question. Oh,
okay.
The second part is that what is the media that's best suitable for use in modeling?
So we did this work using biorelevant media, so passive conditions, and this seemed to work pretty well. Also, our internal experience related to solubility in biorelevant media and see how this compares then with the absorption profiles modeled in vivo points out that this is really a recommendable media to consider.
Okay, thank you. The next question, will it be possible to build or expand the model to incorporate impact of trace levels of stable/low solubility polymorphisms?
So if I understand the question, correct me, Sherlyn, is whether this model could be incorporated to deal also with crystal transform.
The model can include or incorporate the impact of a small amount of stable low solubility polymorphic forms.
Yeah, I guess that the model could be able to do so. It's just a matter of how you formulate it. And yes, I mean, this may, if I understand correctly the question, this may refer to this experiment we did here, where we basically tested the precipitation profile in certain conditions and then we added, we spiked a little bit of powder into this, into another experiment, starting at the same conditions, and we saw how the precipitation got accelerated. So actually the model in its formulation is able to capture the influence that solid particles that exist in the system may have on the kinetics of the process.
Thank you. The next question, was there an interest to use biorelevant dissolution media versus blank buffer for the different concentrations, or can the blank buffer used as well? From Nestle. So essentially, can you just go ahead and use the blank buffer rather than the biorelevant dissolution media?
Yeah, I would actually revert the question and say, could you just do it with FASIF? For us, as I said before, we have a really good experience using biorelevant media to predict absorption when we use PBPK, and for us was the starting point. That's the type of data that we need to generate in order to then link to the in vivo data. The reason why we added the blank buffer in the experimental setting was to address potential differences in the precipitation profiles that could arise from the fact that the compound associates with micelles, and then we start to see things that are fundamentally different. And it was actually very nice to see that at the end of the day, it doesn't matter whether you start from FASIF or you start from blank buffer, you get to similar type of curve patterns. So you get exactly the same curve patterns, and it doesn't seem to have a real difference, and thus the model that we use in one can be used in another. At the end, the only thing that would differ is the value of the 4 parameters that describe the system. So if the question is, could I live with a single set of buffers, then my answer would be yes, but not with blank. I would rather go with FASIF or FESIF, probably not FESIF also due to technical limitations here, but something that really reflects the physiology.
Just to quickly expand on that question, so you would get a different set of parameters if you fitted it to just measurement in the blank buffer versus if you do measurement in FASIF or in FESIF, right?
Absolutely.
So for predictability, the FASIF or FESIF conditions would be the ones that would give you a better prediction for in vivo conditions.
Yes, and we haven't actually tried to model in vivo data with blank buffer data at all. We directly use the FASIF data for that.
Okay, thank you. Do you think that the model can predict the oral absorption for mice, rats, dog, or monkey?
Yes, I would believe so. It may need a little bit of adjustment on the technical setting and to try to reflect conditions in the buffer that are reflective of the physiology of the upper intestine of those different species. So the FASIF is human FASIF, but you could also use different type of animal FASIF if differences in pH or bile acid or phospholipid concentrations are expected relative to the regular, biorelevant medium. But in principle, I think that the same approach could be used to any type of species. And actually, this could be of great interest given the fact that some of these questions start to arise early on in projects before you start to have clinical data.
Okay, thank you. We reached the end of the written questions. Let's see if we have anyone who wants to use the audio. No, I don't see any hands here. Actually, I myself have a question for you, Ruben. I may have missed your presentation earlier while I was trying to monitor the questions here. Your model, can you explain or maybe just provide the rationale or the basis why you can the nucleation term to in order to simplify your model? Because the nucleation, you said this is the slowest step. Yes, the number is the the rate constant is low there. However, if you think about the sequentially for a drug to get into the GI tract to get absorbed, the lowest the step could be rate limiting. If, say, for example, if you don't have a nucleation, you don't have a crystalline growth to occur to begin with. And for some of the compound that has long lag time or relative stable before they start the nucleation growth, and by dropping this term, you probably, I'm not sure, are you missing? I don't know in your example whether you have purposely picked some compound that is relatively stable and take long time to even have a nucleation start. For that case, or your model still apply or still good?
Yeah, I see your point, Shelin. Actually, if you really want to have the, longer explanation on that, I would suggest that you refer to the supplemental information of the article I mentioned before. If you want to get the short one, that's the one that I can give you. Basically, this term is a very slow term. And when then you consider that the change of a compound over time depends on the sum of three different terms. If this one's small and the other ones are big relative to it, this one you can take it out. Of course, this doesn't happen all the time. There are conditions where this term is important, and the conditions where this term is important is where there is no solid available. This is, you only have compound in solution, because in those cases, the amount of solid is 0 here and here, and then these two terms become 0, and that's the only term that exists. And actually, that's the term that then it is responsible for the precipitation to kick off. So if you don't have this term, you cannot explain why a compound that is in a supersaturation state can crash out. You need to have this term in order to explain that there's a certain number of nuclei that need to be formed, and once this is done, then these two terms start to be bigger because this amount of solid starts to be big enough, and then they start to, take over the process. And in practical terms, as long as you have a really minimum amount of solid, this can be 1/1000, your fraction or so, it really depends on each solid, you can be confident enough that this nucleation term can be neglected, and then what you have left is the dissolution and growth. I mean, when you look at it in terms of, the kinetics, that's basically a bit what I mentioned before. At the beginning, you have really a small rate, and this is actually your nucleation rate. You have a very small decrease, and as soon as you have enough amount of solid, and this can be, very limited because you see that the variation in concentration is tiny, then the system accelerates very, very rapidly. And those are actually the dissolution and the growth terms that start to take up.
Okay, thank you. I guess I will spend time to read your paper.
I think that conceptually also gets a little bit dense, so it's a good question.
Let's see. I think we also reach our time too. All right, thank you, Ruben, for answering those questions. If anyone have further questions that we actually run out of time, then you can post the question and go to our Gastroplus user group linking website where you can post your questions, and then your questions will be addressed by our members or the members in Simulation and Plus. So thank you again, Ruben, for the presentation and answering these questions, and thanks Vera also to answer those questions. For the audience, thank you all for your participation. You may now disconnect from the webinar. Thank you, everyone. Bye.
Bye.